In this video, I will demonstrate how to perform proteogenomics analysis of driver events in 2 breast cancer subtypes. I will perform differential protein expression analysis of the RPPA TCGA dataset to identify differentially expressed proteins between luminal A and HER2 breast cancer subtypes. I will then perform a targeted gene expression analysis for genes encoding top 12 most differentially expressed proteins. Then I will construct a genome-wide transcriptomic signature and use it in a CMAP analysis. I will start the analysis by going to the datasets workflow and identifying my dataset of interest. I will focus my search on TCGA datasets. Data types, I will focus on proteomics, and then I will search breast for breast cancer. This search yields 4 datasets. I will analyze the first one, dataset consisting of 937 samples profiled by the reverse phase protein arrays. Click Analyze. This will take me to the landing page for this particular dataset, which has additional information. Then I will follow the Create a Signature link. Signature creation starts with selecting the variable describing the samples that I want to use in the analysis. In our case, the variable that I will use is the PAM50 subtype designation based on the analysis of mRNA data for each sample. So I will select this factor. Then I will select the treatment group, which in my case I will select to be luminal A samples. And then I will select the baseline group that will be used in the comparisons in the differential protein analysis. And I will select HER2 enriched samples to be the baseline. I will now click Create Signature. This process has yielded differential expression analysis of luminal A against HER2 enriched breast cancer subtypes. By default, the procedure selects top 100 probes as differentially expressed. On the landing page for signature created using differential protein expression analysis, we have several panels that display different types of analysis. On the first panel are links for additional pathway, network, and visualization analysis using differentially expressed proteins. The next panel, which I will survey first, contains actual differential expression analysis results for each probe on the microarray. There are 263 probes on this particular RPPA. I can display all of them at once by, say, selecting 1000 here. The probes are ordered by the statistical significance of the differential expression analysis. Immediately, I can notice that ER alpha was the top most statistically significant protein, and it was highly overexpressed in luminal A samples, which is expected. The next 2 most differentially expressed or most statistically significant probes corresponding to HER2 protein, and one of them is phosphorylated version of HER2, the other one is the regular HER2 protein. They were both overexpressed in the HER2 samples, which again is very much expected. If I look at the p-values of the most differentially expressed proteins, I can see the top 12 proteins had all very highly statistically significant differences with p-value less than 10 to minus 10. So in the analysis, subsequent analysis, I will focus on these 12 proteins. To focus on these 12 proteins, I will click on the link that allows me to modify the list of differentially expressed genes or proteins that are used in subsequent analysis. So I will click here. This takes me to iLink's volcano plot app, which allows me to subset genes based on their differential expression and based on their statistical significance. And I will, in this case, use only statistical significance as the threshold, and I will select only proteins that had p-value less than 10 to the minus 10th. And as you can see, 12 proteins match this criteria. I can interact with this with Volcano Plot if I click on interactive and then label the selected 12 proteins as previously seen in the list. The most statistically significant ones were proteins corresponding to the ERBB2 gene and the estrogen receptor alpha. I will now click Analyze, and new results page will be created now that has 12 probes after filtering instead of previous 100, because it is now focused on only 12 top 12 differentially expressed proteins. I will go back now to the analysis results and visualize protein expression data for these 12 probes, and I will do this using Morpheus. So if I click on Morpheus link, it will show me the heat map of the expression levels of these probes in all samples. I will modify slightly this heat map. I'll fit it to Windows so that I can see the patterns, and then I will sort samples based on whether they were HER2 enriched or luminal A subtypes. This now gives me the same heat map that was shown in the paper, in which the proteins, first 4 proteins, are more expressed in the luminal A cancers, and then subsequent 8 proteins are more expressed in the HER2 enriched cancers. I can also look more closely at each of the differentially expressed proteins and look at the distribution of the measurements across 2 different subtypes that we used in the analysis. If I click on this box plot link, now this gives me immediately the direct comparisons of all samples in between 2 different sample types for this particular protein, which in this case is ER alpha. Now I will proceed with using these 12 proteins to analyze corresponding transcriptomic data set, and I will focus only on these proteins to see how this matched analysis transpires in the analysis of gene expression data. In order to do this, I will follow the link for other analysis with selected genes, right? So we have selected these 12 probes, their corresponding genes. And if I click here, I can now again select what type of analysis I want to do. I will go for data sets. And now I will select the corresponding data set, TCGA data set, by focusing my search on TCGA. but now I will use gene expression as the data type, and again I will search for breast. This will give me 5 hits, and I'm interested in these data sets of 1215 RNA-seq samples profiled. So I will click Analyze, and again create a signature. In the same way that I created for RPPA data, I will select PAM50 sample subtypes, and it's going to be luminal A samples as the treatment group and HER2 enriched samples as the baseline group. So the results of this analysis, the results page looks the same, and again the analysis was performed only for 11 genes that corresponding to 12 different protein probes, which we identified in the initial proteomics analysis. It turns out that all of the genes for the proteins analyzed in the first step were also differentially expressed, or statistically significantly differentially expressed, except for EGFR. And if you perhaps remember, in the proteomic analysis, it was actually the phosphorylated version of EGFR that was differentially expressed between the two sample types. So this would indicate that actually there was no change in the transcription of EGFR gene, and the differences in EGFR activity have come from differences in post-translational modifications of this protein, meaning that EGFR is more activated in HER2 breast cancer than it is in luminal A breast cancers. Going back to the analysis results panel, I can again create a heat map of the expression levels for these 11 genes. Fit to window, and again sort by the sample type so that we recreate the figure from the paper. Now I will go back to this same dataset and perform a genome-wide analysis of gene expression data. So instead of focusing on top 11 genes implicated in the proteomic data analysis, I will just analyze all. So I will go back to the datasets workflow, TCGA, gene expression, search for breast, and again now analyze these TCGA datasets of 1215 RNA-seq profiles from the beginning of the dataset workflow, and therefore all genes will be analyzed. Again, I select the factor of interest, which is PAM50 subtypes, and it's going to be luminal A for treatment group and HER2 enriched for baseline group, and creating the signatures. I will now survey overall distribution of differential expressions that we obtain in this analysis by again going to the interactive volcano plot. I can see that most highly differentially expressed genes have a very, very high statistical significance, 10 to the minus 24th. So if I slightly modify these cutoff, but use still very highly significant genes, I will end up with 1399 genes. So I will focus on these genes in the future analysis. First thing, I will again visualize expression data. The processing now takes a little bit more time because I have more, the data matrix is much bigger. Fit the window to get a better survey of what is going on. And then I will hide the IDs just to get things a little bit better organized within my window. And as you can see, even unsupervised clustering based on these top differentially expressed genes clearly separate the two sample types, which is expected always. We have modified slightly this Morpheus that we use in iLinks so that it allows on-the-fly enrichment analysis of clusters of genes. So for example, we can select these cluster of genes that seem to be very highly differentially expressed and with the highest expression in HER2 to samples, and then we can submit this set of genes for, say, enrichment analysis, for example, using Enricher. And if we look at the pathway analysis, it turns out that these genes were related to cell cycle genes. And also in MSigDB hallmarks, we have G2M checkpoint and E2F targets, which usually means that these are genes associated with the proliferation, meaning that in our data set, HER2 proliferate faster than luminal A, which is not unexpected. Going back to the results page, we can now perform connectivity map analysis by going to the connected signatures. Now these are no longer pre-computed as they are for signatures that are already in links, in iLinks. So we will perform a targeted analysis by selecting each of these libraries. Analysis of the connectivity map analysis of corresponding signatures will be initiated on our server. And as they are finished, we can survey the results. Connectivity map analysis for genetic loss of function perturbation signatures. The next one of interest is the genetic perturbation of gain of function signatures. And then finally, chemical perturbation signatures, which is classical connectivity map analysis. And finally, two additional libraries that we used for just as a reference is the disease related signatures and then ENCODE transcription factor binding signatures. We can start with interpretation of these because they are very straightforward. So E2F4 again targets are highly associated with HER2 sample types, which again is has been established by the pathway analysis and usually indicates that these samples are proliferating faster. If we sort by the directionality of the associations, we see that the opposite end ER alpha targets are highly associated with the luminal A samples. which is again obviously reasonable and validates the results of our analysis. In terms of disease signatures, the most highly correlated signatures are again related to the same type of samples, right? It's estrogen receptor negative versus estrogen receptor positive samples, and it shows that basically estrogen receptor positive samples are more similar to luminal A tumors in our analysis because we have negative correlation than estrogen receptor negative tumors, which makes perfect sense. And all the other top hits are all signatures created by comparing some breast cancer subtypes or another. In terms of classical connectivity map analysis aimed at identifying drugs that could reverse a signature, we see that the top 5 signatures were positively correlated with our signatures, which would indicate that they would actually inhibit the HER2 inhibit luminal A signature. We will now perform an additional analysis of the top most correlated perturbagen signatures to see what we can gain in terms of understanding why these signatures were really first 100. We will analyze the most highly correlated signatures, and we will perform group analysis of these signatures. So this is an interface that allows us to specify some specifics about these analysis of 100 most correlated signatures. If by any chance you have a problem with being redirected to the signature group analysis page, make sure to check that pop-ups are not blocked for this particular page. This is the landing page for signature group analysis. It shows the signatures that were analyzed and then results of these analyses. The first thing I will do, I will again open Morpheus heat map to look at these signatures, and again modify slightly. This shows a distribution of compounds that were generating the signatures, correlated signatures. Wortmann Innit seems to have generated quite a few signatures. Cell lines that were used in generating these signatures, and time points. These were all actually 24-hour time points, concentration of the compounds. Finally, we would like to summarize the mechanism of action of chemical perturbagens that yield these highly correlated signatures. We can do that by looking at the Morpheus heat map of drugs by target matrix. Columns in this matrix are target genes or genes of target proteins, and the rows are signatures, and we can actually display the perturbagen generating these signatures. This concludes the workflow that produces the results in the use case 2 in the paper.